Hi everyone, welcome again to the course of structural biology. We are kind of at the end part of the course. We are going through the 11th module, and now, as you know, in the last class we have started the module called protein engineering. Today I will continue from that. So, today the major thing we are going to discuss is strategies of protein engineering. This is actually the core part, the intellectual part. So, to start with, I have kept few questions and answering them so that you will understand how the process is going on. What can be engineered in proteins? So, we have the protein. What actually is our target? So, one of the thing is folding. If you have gone through the course properly, you would understand the importance of folding, the journey, what we talk about, the journey of sequence to function. In between is structure. Structure means folding, and that is why I wrote folding plus structure. So, as I told from the beginning of the course that getting structure, knowing the fold would help us, now you will understand. So, one definitely we will look for is thermodynamic stability. Thermodynamic stability is dealing with the native and unfolded state. If you remember, in the last module of MD simulation, we have discussed about the opening of a protein. The opening of a protein means the opening of a folded state to the unfolded state. Unfolded, folded to unfolded. So, folded state we could also call it native state. So, one of our close look would be how the protein alter from the native to the unfolded state, and can we keep it in the native state, can we enhance the thermodynamic stability. The second one is in a more broader sense, but connected to this is thermal and environmental stability. Under these there would be temperature, pH, solvent, detergent, salt and many more. So, you have a protein, if you all know about, the protein is having a primary sequence which is the protein sequence. Now, then the sequence is made by covalent bond, right. Now, this protein is doing what we call fold, where we get the structure by non-covalent bond. Again, throughout the course we have discussed the non-covalent bonds, they are not as stable as the covalent bond, and these are many in number, like hydrogen bond, salt bridge, you are going to name them, but you could consider as an example which is hydrogen bond. Hydrogen bond is a non-covalent bond which is present in protein in many. So, their state, like they are forming or not forming, have a huge role in showing the stability of the protein. So, one of the biggest target of protein engineering is for stability, because if we get to make the higher stability version, the thermostable version I would say of a protein, it could be used more industrially, because normal protein have Tm melting temperature around 60 degree centigrade, whereas in the industry the industrial process go above 70-75 degree centigrade. So, if you could make a protein stable above 75 degree centigrade, this protein or this enzyme would be industrially suitable, it would be part of the fermenter bioprocess and do not denature. Next thing is function, and one of the aspect of function could be described by binding. Binding is the interaction of the protein with its surrounding, interaction of the protein with other macromolecule, interaction of the protein with substrates, inhibitors, ligands, small molecule, anything. So, how many points are required to bind a molecule with high affinity? A question I posed, which is generally used by people who actually develop graph model and further use those data towards development of machine learning based prediction. What I mean, suppose you have a protein like this, and the protein have 1 amino acid, 2 amino acid, 3 amino acid, 4 amino acid. Then there is a substrate, and the substrate is making non-covalent interaction with the protein. Now, you see that each of these non-covalent interaction would be measured as points, and the number measurement is straightforward and easy. That is one of the reason this is chosen. Definitely it would not say everything, like if I say there are 6 points, I definitely not include the actual chemistry or actual biochemistry here, because all of these interactions are not same. One might be hydrogen bond, other might be salt bridge. Among the hydrogen bond there are different strengths, but the point is the easiest and basic representation for the development of the model. Okay. Then you could further introduce complexity to the basic model. That is how computational model development works. Second thing is definitely catalysis. Binding is happening now. How the enzyme would be converting the substrate to product, that is called catalysis. A different form of binding, binding the transition state of the chemical reaction. So, we will discuss this point a bit little details in structure based drug designing, our last module, but just to talk about. So, when there is a substrate binding to the enzyme, the enzyme starts a reaction. So, what happen? You have A bond B plus C bond D. When the reaction start, the bond between A and B and the bond between C and D breaks. The bond between B and C and A and D start forming. This is the state where bonds are formed, bonds are like forming, and bonds are breaking. This state is called intermediate or transition state. Transition state is very important because binding of a substrate with an enzyme in its transition state shows the intrinsic property of the enzyme. Why intrinsic property is so important? Because if you see sometime nature do the evolution, we will talk about that. Nature makes changes in protein because of many reasons. One of the significant reason is where I research, so I talk about that, is called antimicrobial resistance. So, an enzyme change itself so that it could not bind to the inhibitor, but it could bind its substrate, but the enzyme could not change its intrinsic property. And as I say, I will come back to that on drug designing, because this is a very critical concept in drug designing. So, catalysis engineering is possible, targeting the enzyme, altering its binding to transition state means altering its substrate specificity, altering its choice of substrate and many things else. So, one enzyme, as we all know, the enzymes are in many cases specific. The specificity could be altered by studying the binding of the chemical reaction, how the enzyme is binding to the transition state. But now you could ask me a question, if you understand about the transition state, that transition state, as you say, it is bonds are forming, bonds are breaking. So, it is a hypothetical state, there is no practical existence. So, how you study it? Yes, as I told, I will go in detail in drug designing and show you the techniques how we study an enzyme's binding in its transition state or intermediate of the reaction. So, increased binding to the transition state, increased catalytic rate, altering catalytic rate, a lot of things are possible. What it needs for us to engineer the protein towards catalysis is the knowledge of the catalytic mechanism. We could engineer Kcat, Km. Kcat is the catalytic process, catalytic power, Km is correlated to the affinity. So, now we are talking about factors which contribute to stability. As I talked about hydrophobicity, if you remember, we have discussed about 20 amino acids. In one of the previous modules, we talked about, in the 9th module, the visualization, we talked about pi mole. If you get a PDB downloaded from RCSB protein data bank and load it in the pi mole and go and find the electrostatics, you will get white color that actually refer to the hydrophobic patch or the presence of hydrophobic amino acids. Then you get red, which will show you hydrophilic, the oxygen and negative charge, and then you get blue, which is nitrogen or positive charge. So, in that way, when you look at the electrostatic presentation, you will get the hydrophobic amino acids. The hydrophobic amino acids, valine, isoleucine, leucine, alanine, a lot of them, the aromatic ones, the phenylalanine. So, all of them are contributing to hydrophobicity. Electrostatic interactions are very important for different reasons, especially when you consider protein engineering, which is our current topic. We are more interested in electrostatic interaction because you could measure actually the electrostatic factors. One of them is salt bridge. What is a salt bridge? A salt bridge is, in its stability or in its power, it is in between the covalent bond and non-covalent bond. It is much stronger than a non-covalent bond and weaker than a covalent bond. A salt bridge is formed between aspartate glutamate with arginine, lysine, apart histidine. By introducing a salt bridge, you could enhance the thermostability of a protein. Hydrogen bond as I told, they are not very strong, they are weak bonds, but they are many in number. So, breaking and making of a set of hydrogen bond help protein get more stable, get protein changing its conformation and many things else. Then dipole interaction, you could also look at the dipole interaction and you engineer them. Disulfide bridge is very powerful because it is the only covalent bond which could form in time of physiologic condition. There are two amino acids, cysteine and methionine, who have the sulfur, but methionine does not form a disulfide bond, it is only cysteine. So, when there are two cysteines in a proper position, they could form, but when you are engineering a thermostable protein, you could introduce a disulfide bond, you could design a disulfide bridge. Metal binding of metal makes a domain of the protein more stable. So, what type of metal, which metal, is it coming in di form like di zinc and all. So, these are all consideration. If you consider zinc, they generally bound to like histidine and cysteine. It is mostly in the, it is called zinc finger, where the binding residues are C4 all cysteine, C3H, C2H2, CH3 or H4. So, these are the general scenario. In the same way, there is porphyrin ring which is binding to iron. Here there is a very interesting thing I should tell you, which would be a very good target of protein engineering. If you see a enzyme called cytochrome P450, the enzyme have a porphyrin ring and a iron bound to it. Now, if you take out the iron, if you alter the porphyrin ring so that it could bind to other metals, even other metals which are biologically not seen, you could have carry out catalysis which are not actually feasible in biological condition, but if the enzyme could agree to perform this, you will get the conversion very specific and probably in one step. So, what I am trying to say, like cytochromes which are generally oxygenase type, if you could properly engineer it, you could use these biocatalysis in abiocatalysis to make one step, two step conversion. Very common example, benzene to phenol. You could perform those in very less number of steps and in low cost. So, these are the area where protein engineering could take very critical role. Also reduction of the unfolded state entropy you could work on. How? Any amino acid to proline. Because proline have a considerable rigidity which sometime work on those purposes. Design of thermal and environmental stability, stabilization of alpha helix dipoles. By stabilizing those dipoles, you can increase the stability of the protein in significant amount. You could engineer structural motifs, you could do very interesting creative engineerings, you could make protein oligomerized. A common strategy people use the tetrameric or tetramer forming unit of enzyme P53 and attach that with other protein. So, they tag it, and because it is tag here now, it would bring the same protein hopefully to form a tetramer, which normally form a monomer. So, this type of things are possible. You could also make a protein binding to a DNA which is not binding to DNA by introducing a structural motif which binds to DNA. So, anything you could do. You could introduce salt bridges, as I told. You could introduce residues with higher intrinsic property for their conformational state, like you could replace alanine within a alpha helix, so that you see the change of secondary structure, change of tendency of forming secondary structure. You could introduce disulfide bridge, you could do the reduction of the unfolded state entropy. So, now coming to the protein designing process. Protein designing is the rational design of new protein molecule to design novel activity, behavior or purpose, and to advance basic understanding of protein function. Proteins can be designed from scratch, which is called de novo design, or by making calculated variants of a known protein structure. Suppose already there is a protein, you want to make the protein stable, that is called protein redesign. So, you could do de novo design of a protein with a novel fold, with a combination fold which is not present in nature, they are called de novo designing. But if you take one protein and alter it towards different activity, it is called protein redesign. Generally, the major strategies of protein engineering is divided into two major fields. One is rational protein designing. Rational protein designing is a process which is the actual process which people have adopted. The other one is directed evolution. Directed evolution is something which actually is continuing before we born, before human. When I say we born, I mean the existence of the human community. So, it is what nature used to do to optimize and making sure that the new organism or something coming out have total system balanced and working. So, rational protein designing approaches make protein sequence prediction that will fold to specific structures. These predicted sequences can then be validated experimentally through methods such as peptide synthesis, site directed mutagenesis or artificial gene synthesis. Directed evolution (DE) is a method used in protein engineering that mimics the process of natural selection to steer proteins or nucleic acids toward a user defined goal. As I told, it is a mimic of the process by which nature creates and maintains all the living organisms. Directed evolution consists of subjecting a gene to iterative round of mutagenesis. So, you take a gene and you perform random mutagenesis, creating a library of variants, then you do the selection what you actually want. So, selection expressing those variants and isolating members with the desired function, and then amplification generate a template for the next round. In directed evolution, one of the major challenge is to get a proper selection method of the variants. It can be performed in vivo in living organism like a mouse in the laboratory, ex vivo that means in cells, and in vitro that means you have a gene, you look at the gene's expression, or you take the gene product, the protein, and make an assay out of this. Directed evolution is used both for protein engineering as an alternative to rationally designing modified proteins, as well as studies of fundamental evolutionary principles in controlled laboratory environment. So, these two are what is in practice, the rational designing and directed evolution. But you know, as I continuously talk about, with the innovation and optimization of next generation sequencing technology, biology is looked at a very different way, and as I say continuously to the younger generations, if you see, it is not very old, though it shows that NGS, the first human genome project, was performed in 2003, but if you look out through the journey of every aspects related to NGS and structural biology, it is around 2015 when we start realizing the importance of NGS, when the common scientists start using NGS, because at the initial stage NGS was very costly. A good example is when I joined here, the service says we used to get in India used to charge more than 50,000 to 1 lakh for a bacterial genome sequencing, and currently it is reduced to below 15,000. So, these 5-fold decrease, like now you spend 15,000 and you get the entire information about an organism. That is where the revolution is happening, that is where the biology is changing. Biology is not looked at a gene level, you could have seen all other effects through genomics and transcriptomics, you see the DNA RNA level, through metabolomics you see the small molecules generated. This is giving you the entire picture in front of you. That would make protein engineering, enzyme engineering much easier, I would say much more creativity, much more innovations are coming. So, we talk about the advances of protein structure and function, in advances in high throughput screening. It may greatly expand the abilities of protein engineering, as I explained. Then eventually even unnatural amino acids, we are only talking about 20 amino acids, but why not the modification of amino acids to be incorporated in the protein designing? If it could be happened via newer methods such as expanded genetic code, which is already working, it would allow the encoding novel amino acids in the genetic code, and what more you could do, only with 4 bases, 4 nucleosides, you have 61 codons. Each of one incorporation will make so many, and we could really expand the chemistry. So, with that I am coming to rational protein designing. In rational protein designing, a scientist uses detailed knowledge of the structure and function of a protein to make desired changes towards getting a updated protein. I am keeping it common because updated may be anything. In general, this has the advantage of being inexpensive and technically easy, since size directed mutagenesis methods are well developed. So, what you are doing, you look at the structure, you design it and then make a change, and as I have already explained that whatever you do in protein level, you cannot change the protein level, you have to go and change in the DNA level. However, its major drawback is that detailed structural knowledge of a protein is often unavailable, as the initial description of this course, the challenges, we do not have enough structure. Now you know we have 1,75,000 plus structures, which is coming from around 35 to 40,000 proteins, whereas millions of proteins are now available. Day by day newer proteins are coming. So, that is one major challenge. If you do not have good structural informations, then it is difficult. So, the major drawback is detailed structural knowledge of a protein is often unavailable, and even when available, it can be very difficult to predict the effects of various mutations, since structural information most often provide a static picture of a protein structure. So, that is another thing we have talked about. The majority of the data is based on high resolution X-ray crystallographic data, which represent protein in a static mode. But now for making new mutants, you actually want to get the dynamic data. However, programs such as folding at home and Foldit have utilized crowdsourcing techniques in order to gain insight into the folding motifs of proteins. So, if you had just gone through the module of MD simulation, you will see that making the dynamics of a protein takes huge time, and as I told in the module, we do the folded protein to unfolded. This is studied through simulation. Why? Because you have the folding information, you utilize it to unfold. There is a certainty when something is zipped and it is open up, there is one way around, but if you want to do this, it would take much more computational power, and unfortunately even our supercomputing is not good enough for that. But folding at home by Bijay Pandey is an amazing concept where people are giving access to their own personal computers when they are not using it. Also, folding at home is taking the contribution of gamers. You know, gamers always have high performance computing. So, they are developing games where you make a team and you start playing that, so that you would make number of permutation and combination to achieve an unfolded protein to a folded one. And this initiative is coming up with solutions of protein structures which were not solved before. Computational protein designing algorithms seek to identify novel amino acid sequences that are low in energy when folded to the pre-specified target structure. While the sequence conformation space that needs to be searched in large, the most challenging requirement for computational protein designing is a fast yet accurate energy function that can distinguish optimal sequences from similar suboptimal ones. So, again this is a area where a lot of research is going on, artificial intelligence, machine learning is working a lot, but we still waiting and we need a intertalking between the theoretical studies with the experimental findings. Coming to the concept and history, the goal in rational protein designing is to predict amino acid sequences that will fold to a specific protein structure. Although the number of possible protein sequences is first, as I told, growing exponentially the size of the protein chain, only a subset of them will fold reliably and quickly to one native state. Protein designing involves identifying novel sequences within that subset. The native state of a protein is the conformational free energy minimum for the chain. Now you understand what I am talking about. I have explained this throughout. When we have a unfolded sequence, the way we look at the folding is pushing it towards the lowest energy minima. Thus, protein design is the search for sequences that have the chosen structure as a free energy minimum. In a sense, it is a reverse of protein structure prediction, because when we have protein structure prediction, we have the sequence and we get the structure. Now, we look at how we get a native fold from which sequences. That is why it is a kind of a reverse process. In designing, a tertiary structure is specified first and a sequence that will fold to it is identified. So, we have the tertiary structure and we find a sequence, primary sequence, which will provide or which get from this. Hence, the process is alternatively termed as reverse or inverse folding. So, this is folding, this is inverse folding. Protein design is then an optimization problem using some scoring criteria. An optimized sequence that will fold to the desired structure is chosen. So, here we have the sequence and we want to get the folded information. Here we take a folded structure and get to score to the sequence which will give that. When the first proteins were rationally designed during 1970 and 80s, the sequence for these was optimized manually based on analysis of other known proteins, the sequence compositions, amino acid charges and the geometry of the desired structure. The first designed proteins are attributed to Bernd Gutte, who designed a reduced version of a known catalyst, bovine ribonuclease, and tertiary structures consisting of beta sheets and alpha helices, including a DDT bind to that. Uri and colleagues later designed elastin like fibrous peptides based on rules on sequence composition. Richardson, whom we talked about especially in time of visualization, and co-workers designed a 79 residue protein with no sequence homology to a known protein. In the 1990s, the advent of powerful computers, libraries of amino acid conformations and force fields developed mainly for molecular dynamic simulation enabled the development of structure based computational protein designing tool. If you remember, I told because of this, looking at this contribution in 2013, Nobel Prize was awarded to a field called molecular dynamics. It was the first time, definitely it was given to 3 people, but it was mainly because they have walked through the development of MD simulation and how molecular dynamics simulation contributed to the understanding of protein structure, function and helping in designing of new protein. Following the development of these computational tools, great success has been achieved over the last 30 years in protein designing. The first protein successfully designed completely de novo was done by Stephen Mayo and co-workers in 1997, and shortly after in 1999 Peter Skim and co-workers designed dimer timer tetramer of unnatural right-handed coiled coils. In 2003, David Baker's laboratory designed a full protein to a fold never seen before in nature. So, this is a new fold coming in nature. Later in 2008, Baker's group computationally designed enzyme for 2 different reactions. In 2010, one of the most powerful broadly neutralizing antibodies was isolated from patient serum using a computationally designed protein probe. Due to these and other successes, protein design has become one of the most important tools available for protein engineering. There is great hope that the design of new proteins, small and large, will have uses in biomedicine, bioengineering, and it is just too broad field, but it could be in any field, not only in biology. It could also be in the field of development of electronics, sensors, chemical reactions and what not. So, underlying models of protein structure and function. Protein design programs use computer model of the molecular forces that drive proteins in vivo environments. In order to make the problem tractable, these forces are simplified by protein design models utilizing already available information as well as excellent progress in the field of computation in biology. Although protein design programs vary greatly, they have to address 4 main modeling questions. What is the target structure of the designing? So, the basic target, the target structure. What flexibility is allowed on the target structure? Flexibility imposed, the dynamicity of the model. What or which sequences are included in the search? So, what are the correlation? What are the sequence space? Which force field will be used to score sequences and structures? So, coming to target structure, protein function is heavily dependent on protein structure, as we have talked thoroughly in the course, and rational protein design uses this relationship to design function by designing proteins that have a target structure or fold. Thus, by definition, in rational protein design, the target structure or ensemble of structures must be known beforehand. This contrasts with other forms of protein engineering, such as other one which we told about, directed evolution, where a variety of methods are used to find proteins that achieve a specific function, and with protein structure prediction, where the sequence is known, but the structure is unknown. Here it have to be at least some structural informations are available. Most often the target structure is based on known structure of another protein. So, you might have not the availability of the direct exact structure of that protein, but you need structural information from the related protein, so that you could use that to make a predictive model, 3D model of the target. However, novel folds not seen in nature have been made increasingly possible. So, more and more structures are coming, less and less new folds are available, as it is estimated now that nature have a limited number of folds. Peter Scheme and co-workers designed trimer and tetramer of unnatural coiled coils which had not been seen before in nature. So, if you see, this is GCN4, this is the existing protein, and this is the tetramer RH4 designed by Peter Scheme. You could see that the fold is very different, but these 4 alpha helix you see are the exact alpha helix coming from this protein. The protein top 7 developed in David Baker's lab was designed completely using protein design algorithm to a completely novel fold. So, this protein you are looking at, it is top 7, it is providing a fold which is completely novel, not achieved or not made by nature. Coming to sequence space, as I was talking about, in rational protein design, proteins can be redesigned from the sequence and structure of the known protein or completely from scratch in de novo protein design. In protein redesign, most of the residues in the sequence are maintained as their wild type amino acid, while a few are allowed to mutate and see the effect of those changes. In de novo design, the entire sequence is designed new or as new based on no prior sequence. So, one is de novo and another is redesign, as we talked about. Both de novo design and protein redesign can establish rules on the sequence space, the specific amino acids that are allowed at each mutable residue position. For example, the composition of the surface of the RSC3 probe to select HIV broadly neutralizing antibodies were restricted based on evolutionary data and charge balancing. Many of the earliest attempts on protein design were heavily based on empiric rules on the sequence space. Moreover, the design of fibrous protein usually follow strict rules on the sequence space. Collagen based design protein, for example, are often composed of have the repeating pattern of GlyPro-X, which we see in the collagen fiber. The advent of computational techniques allow designing proteins with no human intervention in sequence selection. The whole sequence selection thing is done by automated computation. In protein designing, the target structure of the protein are known, right. So, what I am saying by that, the static information of the 3D structure is known, but now we are talking about a dynamic model. However, a rational protein design approach must model some flexibility on the target structure in order to increase the number of sequences that can be designed for that structure and to minimize the chance of a sequence folding to a different structure. So, the introduction of dynamicity would be really critical for optimizing the designing approach. For example, in a protein redesign of one small amino acid such as alanine in the tightly packed core of a protein, very few mutants would be predicted by a rational design approach to fold to the target sequence if the surrounding side chains are not allowed to be repacked. So, what I am trying to say, suppose you have a small amino acid and this is your structural periphery. So, you cannot allow to design a bigger amino acid because and in this static environment there is a possibility of steric clash, because the environment where the small amino acid alanine or glycine were existing, a arginine in theory is not possible, but if the model could be showing dynamicity, then we could understand what type of or which amino acid could be packed there. Thus, an essential parameter of any design process is the amount of flexibility or the dynamicity allowed for both the side chains and the backbone. If we have a dynamic model, the designing would be much more accurate. In the simplest model, the protein backbone is kept rigid, while some of the protein side chains are allowed to change conformations. However, side chains can have many degrees of freedom in their bond lengths, bond angles, and the chi dihedral angle, the side chain dihedral angles, which we have talked about before. So, what is the problem? You have phi and psi, you work on phi and psi, you calculate, you get Ramachandran plot, but when you are allowing to consider the chi's, there are so many chi's that your calculation would be very, very complicated. You need to simplify the case, and to simplify this phase, protein design method use rotamer libraries. What is a rotamer library? Rotamer libraries are libraries that assume ideal values for bond lengths and bond angles, while restricting chi dihedral angle to a few observed low energy conformation termed as rotamers. So, some of the rotamers which are selected on different criteria are there, and you cannot change beyond that to make the process simplified. Rotamer libraries describe rotamers based on an analysis of many protein structures. So, backbone independent rotamer libraries describe all rotamer. So, what is this rotamer libraries? They take similar structures and see the possibility of rotamer position of an amino acid, and the statistical consideration choose the winners, and they are in the library, whereas there are backbone independent rotamer library which have all of them. Backbone dependent rotamer libraries in contrast describe the rotamers as how likely they are appear depending on protein backbone arrangement around the side chain. The rotamers described by rotamer libraries are usually regions in space. Most protein design programs use one conformation, that is the modal value for rotamer dihedral in space, or several points in the region described by the rotamer. The Osprey protein design program in contrast model the entire continuous region. So, if you see, you would see that there is a rotamer. So, the common protein design programs use rotamer libraries to simplify the conformational space of protein side chains. If you look at this animation loop through all the rotamers of the isoleucine amino acid based on the penultimate rotamer library, which is one of the premier library at the initial stage of protein designing provided by Richardson. Coming to energy function, rational protein design techniques must be able to discriminate sequences that will be stable under the target fold from those that would prefer other low energy competing states. Thus, the protein design requires accurate energy function that can rank and score sequences by how well they fold to the target structure. At the same time, however, these energy functions must consider the computational challenges behind protein designing. So, more accurate it would be, more complicated the calculations are, more time consuming it would be. So, that is the challenges and accuracy and optimization is required. One of the most challenging requirements for successful design is an energy function that is both accurate and simple for computational calculations. The most accurate energy functions are those based on quantum mechanical simulations. If you remember, I talked about when I was talking about MD simulation. In MD simulation, you are providing classical physics, whereas in quantum mechanics, what you are doing is the electronic level calculation, which is even sometime more accurate than the experiment, like real life experiment. However, such simulations are too slow, as I told, it is time demanding and computation demanding. It is typically impractical for protein designing, considering the big size of a protein as a macromolecule. Instead, many protein design algorithms use either physics based energy function adapted from molecular mechanics simulation programs, knowledge based energy function, or a hybrid. The trend has been toward using more physics based potential energy functions. Physics based energy functions such as AMBER, CHARM are typically derived from quantum mechanical simulations and experimental data from thermodynamics, crystallography and spectroscopy. So, it is a good estimation, good optimized. These energy functions typically simplify physical energy function and make them pairwise decomposable, meaning that the total energy of the protein conformation can be calculated by adding the pairwise energy between each atom pair, which makes them attractive for optimization algorithms. Physics based energy functions typically model an attractive repulsive Lennard-Jones term before atoms and a pairwise electrostatic Coulombic term between the non-bonded atoms. Statistical potentials, in contrast to physics based potentials, have the advantage of being fast to compute, of accounting implicitly of complex effects, and being less sensitive to small changes in the protein structure. These energy functions are based on deriving energy values from frequency of appearance on a structural database. Protein design, however, has requirements that can sometimes be limited in molecular mechanical force fields. Molecular mechanics force fields, which have been used mostly in molecular dynamics simulations, which we have discussed in detail, are optimized for the simulation of single sequences, but protein design searches through many conformation of many sequences. Thus, molecular mechanics force fields must be tailored to protein design. In practice, protein design energy functions often incorporate both statistical terms and physics based terms. For example, the Rosetta energy function, one of the most used energy function, incorporate physics based energy term originating in the CHARM energy function and statistical energy terms such as rotamer probability and knowledge based electrostatics. Typically, energy functions are highly customized between laboratories and specifically tailored for every design. So, we are trying to develop a lot of force fields, we are trying to develop a lot of matching, but depending on cases you have to make modifications which are protein specific or target specific. So, here is a comparison of various potential energy functions taking quantum mechanics, molecular mechanics with explicit solvent, with continuum solvent and heuristic. So, what is explicit solvent? If you remember, I talked about this in simulation. When you consider, let us say, water model, you could consider them as discrete water molecules, as individual existent water molecule. These are explicit solvent, and when you consider them as a continuous solvent with average property, then it comes to continuum solvent. So, in quantum mechanics there will be Coulomb's law, Schrodinger equation. For bond you have to use bond length, which is stretching, bond angle, which is bending, torsion angle or dihedral angle. For non-bonded you use Lennard-Jones potential and Coulomb's law. In molecular mechanics, this is molecular mechanics with explicit with continuum solvent, bonded and non-bonded are same. For continuum solvation you have to calculate the surface area and PB equation. For heuristic you have to take the conformational strain, the statistical terms which will come from different areas and which could be distributed from the information of the different areas. You have to consider the steric classes, you have to consider complementarity, you have to consider chemical complementarity which is charge patterning and hydrogen bond distribution. Protein folding design or protein design as an optimization problem. As we know, the goal of protein design is to find a protein sequence that will fold to a target structure. A protein design algorithm must thus search all the conformation of each sequence with respect to the target fold and rank sequences according to the lowest energy conformation of each one as determined by the protein design energy function. Thus, a typical input to the protein design algorithm is the target fold, the sequence space, the structural flexibility and the energy function, while the output is one or more sequences that are predicted to fold stably to the target structure. So, we have the target structure, we have the sequence space, we have the structural flexibility and energy function. Challenges: water makes up most of the molecules surrounding protein and is the main driver of the protein structure. If you look at the protein structure, we have shown you in the molecular visualization, you have seen the red dots which are water. Thus, modeling the interaction between water and protein is vital in protein design. The number of water molecules that interact with the protein at any given time is huge and each one has a large number of degrees of freedom and interaction partners. Instead, protein design programs model most of such water molecules as a continuum, modeling both the hydrophobic and solvation polarization. Individual water molecules can sometimes have a crucial structural role in the core of the protein and in protein and protein ligand interaction. Failing to model such waters can result in misprediction of the optimal sequence of a protein interface. As an alternative, water molecules can be added to rotamers. So, if you understand the problem, there is a protein, it have a lot of water molecule inside and outside the protein. If you want to consider all of this water molecule individually, your calculation would be hugely enhanced. So, what you do, you take it as a continuum effect, but there are some water molecules which have catalytic role or binding role. If you could identify those water molecule through previous studies, that is the best situation. If not, then there is a risk of those things taking part in the misprediction. An alternative is there to take the water and include in the rotamer library those water molecules. The number of candidate protein sequences, however, grows exponentially with the number of protein residues. For example, there are 20 to the power 100 protein sequences of length 100 amino acids. Furthermore, even if amino acid side chain conformations are limited to few rotamers, this results in an exponential number of conformations for each sequence. Thus, in our 100 residue protein and assuming that each amino acid take exactly 10 rotamers, a search algorithm that searches this space will have to search over 200 to the power 100 protein conformation. So, this number says what is the problem or what is the computational challenge. The most common energy function can be decomposed into pairwise terms between rotamers and amino acid types, which casts the problem as a combinatorial one and powerful optimization algorithm can be used to solve it. In those cases, the total energy of each conformation belonging to each sequence can be formulated as a sum of individual and pairwise terms between residue positions. If a designer is interested only in the best sequence, the protein design algorithm only requires the lowest energy conformation of the lowest energy sequence. So, if you fix them, then you could get rid of a lot of calculations. In these cases, the amino acid identity of each rotamer can be ignored and all rotamers belonging to different amino acids can be treated as same to reduce the computation. If you consider Ri be a rotamer at the residue position I in the protein chain and Eri the potential energy between the internal atoms of the rotamer, let Rj be the potential energy between Ri and rotamer Rj at residue position j. Then you define the optimization problem as one of finding the conformation of minimum energy Et equal to summation of Ei Ri plus I not equal to J Eij Ri Ij summation. This minimization is considered as a NP hard problem. NP hard problem is where we consider the macromolecule the forming a polynomial which is working as a hard one. But even though the class of problem is NP hard, in practice many instances of protein design can be solved exactly or optimized satisfactorily through heuristic methods. So, if you look at here these animations, it illustrates the complexity of a protein design search. By the way, this is taken from a structure of myoglobin. This typically compares all the rotamer conformation from all possible mutation at all residues. In this example, here we are taking phenyl 36 and his 106, they are allowed to mutate to respectively the amino acid tyrosine and asparagine. Phenylalanine and tyrosine have 4 rotamers each in the rotamer library, while asparagine and histidine have 7 and 8 rotamer in the library. As I told, this is Richardson's penultimate rotamer library. So, the animation loop through 4 for phenylalanine, 4 for tyrosine. So, 4 for phenylalanine, 4 for tyrosine and then 7 for asparagine and 8 for histidine. So, 8 into 15 equal to 120 possibilities. Coming to the algorithms, several algorithms have been developed specifically for the protein design problems. These algorithms can be divided into 2 broad classes: exact algorithms such as dead end elimination that lack runtime guarantees but guarantee the quality of the solution, and heuristic algorithms such as Monte Carlo, which we have explained in time of MD simulation, that are faster than exact algorithms but have no guarantees on the optimality of the results. So, 2 type of algorithms: exact algorithms takes a lot of time but give you accurate result, and other ones heuristic algorithms such as Monte Carlo, it would be time saving but not guarantee of the optimization. Exact algorithms guarantee that the optimization process produce the optimal according to the protein design model. Thus, if the prediction of exact algorithms fail when these are experimentally validated, then the source of error can be attributed to energy function that allowed flexibility, the sequence space or the target structure. Most of the algorithm used or mentioned here for protein designing purpose address only the most basic formulation of the protein design problem, the equation taken which we have already discussed for measuring the minimum energy Et. When the optimization goal changes because designers introduce improvements and extension to the protein design model, such as improvement to the structural flexibility allowed or protein backbone flexibility, or including sophisticated energy terms. Many of the extension on protein design that improve modeling are built at top of this algorithm. For example, Rosetta design incorporates sophisticated energy terms and backbone flexibility using Monte Carlo as the underlying optimizing algorithm. Osprey's algorithm built on the dead end elimination algorithm, and Esther to incorporate continuous backbone and side chain movements. Thus, these algorithms provide a good perspective of the different kind of algorithms available for protein design. Good to mention, though I will talk about that little details later. In July 2020, scientists reported the development of an AI based process using genome databases for evolution based designing of novel proteins. They use deep learning to identify design rules. We will talk about that in details. So, coming to wet lab experiments, what we had done now, we have designed a protein. Now, as you know, when we do the wet lab experiment, you cannot do that on protein. So, we have to go to the DNA. As we all know, we have 20 amino acids, and for these 20 amino acids there are 64 codons, 61 specifically for these 20 amino acids and 3 is stop codon. This is the codon library where you see the relations, the relation between the amino acids which are here. These are stop, histidine and with the nucleosides. So, further we will work on modification of the DNA, because in biology the only change which goes further is DNA, as we have discussed in previous classes. So, we have to perform mutagenesis. As we all know now, mutagenesis, the change in the DNA sequence. It might be point mutation, it might be large modifications. I talked about them while discussing protein structure. So, this is a point mutation if you want to cut this part. So, this is deletion. If you want to incorporate a new part, you incorporate an extra part, this is insertion. So, in that way large modifications could be done. So, point mutations are directed mutagenesis, substitution, change of one nucleotide, insertion could also be pointed, gaining one additional nucleotide, deletion, loss of one nucleotide, they are coming under point mutation. So, we will discuss different type of point mutations within the coding sequence and how that incorporated towards mutagenesis and their effect. So, today we have discussed about basic principles about what we target towards protein engineering, how we plan, what is our goal, what type of things we want to see change, and the major process of development of protein designing. One is rational designing, where we go and do our designing, whereas another is directed evolution, where we mimic the nature. We have discussed about how this rational designing happened, and in the next class we will talk about the further consequence of those protein mutation on rational designing, and we will start the other one, the directed evolution. Thank you very much for listening. Thanks.